Good morning friends. Today we are going to discuss about plasma membrane. Plasma membrane. So already we have discussed what is plasma membrane in previous class, cell theory. Plasma membrane is the living covering of a plant and animal cell, and it is the only covering in case of outer covering. Only in case of animal cell it is the outermost limiting, but it is surrounded by cell wall in case of plants. So today we will discuss about the structure of plasma membrane, how plasma membrane is made of and how it looks under microscope. To describe the structure of plasma membrane, one special theory is given, model known as fluid mosaic model. So many models are given, but fluid mosaic model is important. Fluid mosaic model. Fluid mosaic model. Why this model is called as a fluid mosaic model, I will explain. Different other models are also there. These are not required at this time. I will also tell later. So fluid mosaic model. This model is given in 1972. This to describe the structure of cell membrane. Fluid mosaic model is given in this year by two scientists. One is Singer, another is Nicholson. Singer and Nicholson, two scientists. Singer and Nicholson together given this theory. You can easily remember two scientists, easily 72, not 74. Sometimes it may confuse in MCQ type of question in which year this is given. So two scientists and given in the 1972 fluid mosaic model. According to Singer-Nicholson's fluid mosaic model, the plasma membrane or cell membrane is made of two important chemicals, major chemicals. One is lipid, number one is lipid or fat. जो lipid हम बोलते हैं fat को, clear? Lipids and protein. These are the two major components, lipid and protein. Maximum. Other chemicals are also present, but lipid and protein are maximum. So इसलिए बोला जाता है lipoprotein. Fluid mosaic model. Plasma according to fluid mosaic model, the plasma membrane is made of protein and lipid. Lipoprotein in nature, protein or lipid in nature. So one by one we will discuss what is lipid, which type of lipid present and which type of protein present in the plasma membrane. First come to lipids. If you come to the lipid portion of a plasma membrane, a special lipid is present in the plasma membrane, and that lipid is known as a phospholipid. Phosphorylated lipid, phosphate containing lipid. The lipid are fat which contain phosphate. जैसे हमारा नॉर्मल फैट है, घर में यूज करते हैं, हमारे बॉडी के अंदर भी फैट है, तो वही फैट के साथ अगर फॉस्फेट कनेक्टेड हो जाएगा उसको बोलेंगे फॉस्फोलिपिड्स. तो लिपिड्स स्पेशल टाइप ऑफ लिपिड इज प्रेजेंट इन द सेल मेम्ब्रेन. दिस क्वेश्चन फ्रीक्वेंटली आस्क्ड इन नीट as well as other entrance exam. So you have to remember the lipid found in the plasma membrane is known as a phospholipid, also known as a phosphoglyceride. Phospholipid or phosphoglyceride. Phospholipid or phosphoglyceride. Sometimes it is also called as lecithin. Actually, these phospholipids are different type. One is lecithin, sphingomyelin. Different type are there. Lecithin is the most common, found inside the cell membrane or plasma membrane. This question also comes. Which phospholipid is found? That is lecithin or phospholipid or phosphoglyceride. If you come to the structure of this lipid, phospholipid or lecithin or phosphoglyceride, it is showing two important parts. Each phospholipid contains two parts. This is called a head. This portion, this part is called head. This is the head. Upper part is called head of the phospholipid. This is head, and this part is called as a tail. So two parts are present, head and tail. But if you count the number the head is only one, but number of tail is 2. You can easily visualize here 2 tail. Each phospholipid, this is the picture of a phospholipid. Each phospholipid contains one head and 2 tail. Number varies. Another important thing comes. This difference also asks in NEEt as well as other entrants. How many head, how many tail are there? Another important thing is their chemical nature. If you come to the head and tail portion, the head, the important characteristic of a head, the head can react with water. Water molecule can react with this head portion. So that's why it is a hydrophilic. Hydrophilic means water loving. So head portion is hydrophilic in nature. Hydrophilic. It is hydrophilic in nature. Hydro means water, philic means loving. So hydrophilic or water soluble. It is water loving portion, or not water soluble. We can say water hydrophilic. Hydrophilic or water loving part. But the tail portion is hydrophobic. Hydrophobic. So this is very important. Hydro means water, phobic means hating. Philic means loving. So water loving part is the head, water hating part is the tail. Why? The head is different from tail in this hydrophilic or hydrophobic in respect of water loving. It is observed this tail part of the phospholipid is made of saturated hydrocarbons. Saturated hydrocarbons. Saturated hydrocarbon. What is saturated hydrocarbon? You have already read what is a hydrocarbon, compounds of hydrogen and carbon. But saturated means there is no double bond, only single bond are found, no double bond. When double bond are absent, that is called saturated. And a saturated hydrocarbon generally not react with any chemical. तो इसलिए इसका नाम हो गया हाइड्रोफोबिक. तो हाइड्रोफोबिक, why? Tail is hydrophobic. 2 tail are there and the tail is hydrophobic because it contains saturated hydrocarbon. So that's why tail portion cannot react with water, but head portion can react with water. So head is hydrophilic, but tail is hydrophobic in nature. Due to this reason, as the same, this phospholipid, entire is phospholipid, but this part is head, this part is tail. Due to different nature, one part is hydrophilic, another part is hydrophobic. The phospholipid are known as amphipathic in nature. तो phospholipid को क्या बोला जाता है? Amphipathic molecule. Amphipathic molecule. Just like you have read amphibians. Frog live in water as well as in soil. Similarly, here amphi means both. Here water loving part, hydrophilic is there, and water hating part is also there, hydrophobic. So philic and phobic, 2 parts are present. So that's what is called amphipathic molecule. So phospholipid is an amphipathic molecule, and it is found in the cell membrane or plasma membrane, irrespective of any species, any organism you can take. Cell membrane of a bacteria, cell membrane of a protozoa, any organism you enter, you find this type of phospholipid, and all the phospholipids are amphipathic in nature. Clear? This is all about your characteristic of phospholipid. You have to remember why tail is hydrophobic, because it is made of saturated hydrocarbons. No double bond is found, only single bond. So that's why it cannot react. So now come to the detail structure, how these phospholipids are arranged in the cell membrane. If you look at the cell membrane, the phospholipids are arranged in a bilayer. The phospholipids, the lipids which are present, the phospholipids are arranged in a bilayer. Bi means 2. They are arranged in a bilayer. Bilayer means double layer. Yes, if you look at a cell, this is the cell, any cell, animal cell. The outer covering is the cell membrane or plasma membrane. This is the cell membrane or plasma membrane. If you look at the phospholipids are arranged like this. I am showing the phospholipids are arranged like this. It is in bilayer, just like a sandwich, 2 layer, and in the middle something is given. So here you can see this is the enlarged view of a cell membrane. If you look at the cell membrane under microscope, this cell membrane give appearance like this. Okay. Now this portion is expanded. Actually it is a single membrane but contain two lipids. Membrane is single but chemical occur in double layer. So इसलिए क्या लिखा? Phospholipid bilayer. So now we will discuss this phospholipid bilayer in expanded form. Now come to out. I am showing outside how the phospholipids are arranged. If you look, the phospholipid, each phospholipid contains one hydrophilic head, two hydrophobic tail. Clearly visible. One hydrophilic head, two hydrophobic tail. You can easily mark here. One hydrophilic head, two hydrophobic tail. And they are arranged like this. The phospholipids are arranged in a bilayer. And the arrangement is tail to tail, not head to head. You can see tail to tail. One layer's tail is attached to another layer's tail. Tail to tail arrangement, not head to head. Why this type of arrangement takes place? One question may arise in your mind. They can be arranged like this. They may be arranged like this. But in the diagram I am not shown like this. The arrangement can be possible like this, and another arrangement, another possibility. They may be arranged like this, head to head. Head to head, their head are together. Here tail are together. But this type of arrangement not takes place. Why this type of arrangement not takes place? If you come to the cell membrane, this is the cell membrane. Cell membrane having two surfaces. This is the outer portion, this is the inner portion, inner surface. One side is inner. Inside the cytoplasm, it is facing towards cytoplasm, it is facing outside. So two face. If you look at the two face, water is present at both sides. Water is present inside the cell as well as outside the cell, because when you are taking water, water is present outside the cell as well as some water enters, maximum water enters inside the cell also. So water is found in both sides. So that's why water always comes in contact with the inner as well as outer surface. And just few minutes before I had told you, in phospholipids or lipid present in the cell membrane, there is two portions, head and tail. What is the characteristic of head? Head is hydrophilic, tail is hydrophobic. So hydrophobic means water hating, tail are water hating. But water is present on both sides. As water present in both sides, they create aqueous environment. Aqueous means water. So outside is aqueous environment, inside is also aqueous environment. As both sides are aqueous environment, only hydrophilic or water loving part can remain to that side. Which side contain water? So as outer side is aqueous, water, inner side is also aqueous. So outer and inner side, both sides, only exposed to the head. Head can be exposed to that side. Tail cannot be. Why cannot be tail? Because tail is hydrophobic. If tail part will expose to this aqueous environment, then structure will destabilize. Clear? So water hating part cannot face to out as well as inside. So that's why in this diagram you can see only head is facing outside as well as inside. This is the inner face. You can see here. This is the inner face. Head is facing towards inner side as well as upper layers. Head is also facing towards outer side. But tail is protected. So to protect the tail from the aqueous environment, this type of situation takes place. But if you imagine this type of situation, what will happen? Outer side there is water, inner side is also there water, but outer side there is no problem because head is face. Here head is facing, but here tail is hydrophobic, so that's why this type of situation cannot be. So similarly, if you come to this water is present outside, water is present inner side. So inner side, both sides, tail is exposed, so that's why this type of structure is not possible. So only possibility, as both sides contain water, hydrophilic, this hydrophilic head can react with this side water, this hydrophilic head can react with this side water. So that's why always hydrophilic head is exposed to out and a tail is protected from the aqueous environment. Tail is protected from the aqueous environment. This is phospholipid bilayer. This is a phospholipid bilayer. So phospholipids are arranged in a bilayer, phospholipid bilayer. This is the phospholipid bilayer. Phospholipid. This given is phospholipid bilayer. Bilayer means two layer. So phospholipids are arranged in two layer, bilayer, phospholipid bilayer. So reason clear why this type of arrangement is possible: to protect the tail from the aqueous environment, because both sides contain water. This upper side, outer side contain water, inner side. This is the section. We are observing a section. So this is outside, this is inside. So I can write here. This is outside of cell membrane. Outer side of cell membrane. This is inside. Inside of cell membrane. Both sides contain water. So that's why to get protected from the water, this type of situation takes place. But it is observed, in addition to this lipid, another most important chemical is present, which is called protein. So in the fluid mosaic model, I will explain why this model is called fluid mosaic model at the end. But you remember, here two chemicals are important. One is protein, another is lipid. So lipid part is clear. Second part is protein. Next most important chemical is protein. So protein are also present in the cell membrane according to Singer and Nicholson. But they divided or categorized protein into two types. Two types of protein are found. One is extrinsic or peripheral protein. Extrinsic or peripheral protein. Another is intrinsic or integral protein. Integral. Two types of protein are there. Peripheral or extrinsic, integral or intrinsic. So now we will discuss what is extrinsic protein, what is intrinsic or integral protein. It is observed. This is the phospholipid bilayer. In this bilayer, some proteins are present on the surface. Two surfaces are there. You can see here. This is the outer surface. I have shown outer surface of the plasma membrane. This is the inner surface. What happens? Sometimes protein get attached to the outer surface. This is the protein. And sometimes the proteins are attached to the inner surface. You can see in this diagram. Three proteins I have shown. One protein on outer surface and two proteins on the inner surface. And when the proteins are found attached to the surface of the lipid bilayer, on the surface of lipid bilayer, whatever the surface may be outer surface or inner surface. When lipids are attached to the surface of phospholipid bilayer, these are called as peripheral or extrinsic protein. First, extrinsic or peripheral protein. Extrinsic or peripheral protein. Extrinsic or peripheral protein. So what is extrinsic or peripheral protein? Those proteins which are found on the surface of lipid bilayer. Surface of lipid bilayer are known as extrinsic or peripheral protein. But what about the second category? Now I am moving towards that second category of protein. What is written in second category of protein? It is intrinsic or integral. The intrinsic or integral protein are that type of protein which are buried partially or completely in the lipid bilayer. Buried or embedded in the lipid bilayer partially or completely. Now I am showing diagrammatically. Sometimes the proteins are embedded completely or partially. You can see here. Sometimes you can find the proteins are embedded partially or completely. Previously, these three proteins we have shown, they are found on the surface, only surface. So surface proteins are called extrinsic or peripheral. But now you cannot tell it as a surface protein. This is not surface protein, it is embedded in the lipid. In one side, lipid is absent, lipid is present in bilayer, but here lipid is present in monolayer. Only one lipid is present in the lower side, but upper side it is filled with protein. Means protein is embedded or protein is buried inside. Similarly, this type of embedment is called as, it is partial embedment. But sometimes the protein are embedded completely like this, like this. So you can see in this diagram two types of embedded protein I have shown. Some proteins are embedded completely and some proteins are embedded partially. You can see one protein is embedded partially, another protein is embedded completely. And when the proteins are embedded partially or completely, when proteins are embedded partially or completely, that is called integral or intrinsic protein. Integral or intrinsic protein. So two type of protein are there. Protein according to arrangement. You can remember two type of protein. One is peripheral protein, another is integral protein. The protein which are present on the surface, sometimes difference comes. What is the difference between peripheral and integral protein? Those proteins which are present on the surface are called as extrinsic or peripheral protein, and those proteins which are embedded, buried, deep, partially or completely. Here you can see it is partially, only buried in one side, but it is buried in both sides. Both side lipid are absent here. In this place, lipid are completely absent. So it is completely buried or wholly buried. So this type of protein are called as integral or intrinsic protein. And if you compare, another difference is the age of extraction. If you compare these two protein, now tell me, I am asking one question. Out of this type of protein, one extrinsic protein is there, one intrinsic. Which type of protein you can remove easily? You can extract easily. So definitely, answer is extrinsic protein, because extrinsic protein are present on the surface, they are not buried inside. So extrinsic protein can be removed easily from the surface. So extraction is easy in case of extrinsic protein. But in case of intrinsic protein, as they are buried, we cannot remove them easily. So that's why they are difficult to remove. So extrinsic protein are easy to remove. Extrinsic are easy to remove because present on the surface. Intrinsic protein are difficult to remove because they are buried inside. But if you come to their abundance, maximum protein in the cell membrane are intrinsic type, 70%. Out of total protein, 70% are of this category, but extrinsic are only 30%, very less. So extrinsic protein are very less as compared to the integral or intrinsic protein. So this is all about type of protein present. But if you come to this integral protein, I had told you, integral protein are two types. Some integral protein are partly embedded, you can see in the diagram. Some integral protein are completely embedded. And when the protein is completely embedded, this type of protein, you can see the part of the protein in both surface. In this surface this is visible, in this surface also this is visible. In the inner surface it is visible, in the outer surface. But in this type of protein, you find only it is visible in outer surface, or either in outer surface or in inner surface, but not in both. In this protein, only outer is visible, but inner surface is not visible because it is not completely embedded. So integral protein are two types. One is partly embedded, one face is visible, another is completely embedded. Two faces are visible, both sides are visible. Both sides visible means you have to see from both sides. Here side is visible, but from the lateral side, not from this surface, lower surface. When a protein embedded completely, it's both surface is visible, outer and inner. Here it is a transverse section, that's why this part is visible. But in a complete three-dimensional structure, this part is not visible because it is occupied by lipids in the lower side. So in this protein, if you see, in this integral protein, both sides is visible because it is completely embedded. And this type of protein, the type of integral protein which is completely embedded, it is called as a transmembrane protein. Transmembrane or tunnel protein. Transmembrane or tunnel protein. Sometimes question comes, so what is a transmembrane or tunnel protein? the type of protein which is completely embedded and both sides are visible. Both surface, it protrude in both sides, outer side and inner side, outer surface and inner surface. That is called transmembrane or tunnel protein. And this transmembrane or tunnel protein are very special. The transmembrane or tunnel protein sometimes occur single. Here I have shown single transmembrane protein, or sometimes they occur in group. Sometimes they occur in group. The transmembrane protein sometimes occur single or sometimes occur in a group. The transmembrane protein are very special. They occur in group. Sometimes the transmembrane protein occur in a group like this. I am showing group of transmembrane protein. 5 to 6 transmembrane protein like this. Sometimes transmembrane protein occur in a group. Here single transmembrane protein. Here group of transmembrane protein. And when transmembrane protein present, transmembrane protein having a special function. This is the phospholipid bilayer. In the phospholipid bilayer, black phospholipid bilayer, this protein are continuing. This sometimes the integral protein, this transmembrane protein occur in a group or single to form a channel. Sometimes there is a formation of a channel, tube-like structure. A tube is created, pipeline. A pipe-like structure is created, tube-like structure. This structure is called as a channel. This is called as a channel protein. Channel protein. So what is channel protein? You have to remember channel protein. Sometimes question comes. Channel protein, how channel protein is formed? Channel protein is a channel-like structure. It is a cylindrical tube-like structure which is formed by either single transmembrane protein, एक transmembrane protein बना सकता है या ग्रुप भी आ सकता है. यहाँ पर मैं ग्रुप दिखाया, लेकिन एक ट्रांसमेम्ब्रेन प्रोटीन भी ऐसा बना सकता है. तो चैनल प्रोटीन, channel is made of single or multiple, maybe single or multiple transmembrane protein. But what is the function of this channel protein? This channel protein helps in movement of water and ions from the outer to inner as well as inner to outer. So water and ions, this water and ion cannot pass through the lipids, because if you see here, these are filled with lipid. This is the phospholipid bilayer, and the lipid, in case of lipid, the head is hydrophilic. Already you have discussed. The head part is hydrophilic. But tail part is hydrophobic. As tail part is hydrophobic, this tail part is hydrophobic. It hates water. So when water try to enter from out to inner side, head will not give any problem, any resistance, but the tail will not allow, because tail part is hydrophobic. So when water will try to enter, this tail will not allow to cross the membrane. So that's why for water and ions, for movement of water and ions, water molecule and ion, pass from one side to other side. Water and ion can pass from one side to other side through this channel. So that's why in the cell membrane for water you find water channel, and for ion you find ion channel. So different ion channels are present in the cell membrane to allow the passage of ions through the cell membrane. Clear? So this is all about your different type of protein. Extrinsic protein, intrinsic protein, what is channel protein. But important thing here, you have to remember, in addition to this protein and lipid, some other chemicals are also present. In case of human erythrocyte, RBC, it is observed, in case of human RBC, also called as erythrocyte, in case of human RBC, erythrocyte, the amount of protein which is present, it is 40%. Amount of lipid which is present, lipid is 40% and protein is 52%. So total contributing, lipid and protein contributing 92%, but the rest are the carbohydrates. So in addition to these two chemicals, other chemicals are present. These are called as carbohydrates. So in the plasma membrane, you find carbohydrate also. Carbohydrate means sugar type of chemical, not hydrocarbon. Hydrocarbon is different. Hydrocarbon is a general name. Carbohydrate is a specific name. So carbohydrates are present, and these carbohydrates are present mainly on the outer surface. This is the cell membrane on the outer surface, and this carbohydrate or sugar are mainly oligosaccharides. I will explain you what is oligosaccharide. Medium carbohydrate. Carbohydrates are divided into three categories. One is monosaccharide, oligosaccharide, and polysaccharide. Polysaccharide are large, oligosaccharides are medium, and monosaccharide are smallest. But these are oligosaccharides. I will explain later in another chapter what is oligosaccharide. So what happens on the extrinsic protein? The protein which are present on the surface proteins are called extrinsic. These are the extrinsic protein. So extrinsic or peripheral protein. With extrinsic or peripheral protein, some oligosaccharides are attached. This is oligosaccharides as shown. Oligosaccharide is attached, and the attachment bond is covalent. There is a covalent bond, covalent linkage. Already you have read in lower classes different type of bond, chemical bonding, ionic bond, covalent bond. Due to covalent bond, the protein, this is a sugar, and this is the extrinsic protein. Extrinsic protein and sugar get connected, and it form a structure known as a glycoprotein. This structure is called a glycoprotein. But here protein is present, but no sugar. So this is called only protein, but this is called a glycoprotein. So this structure is called as a glycoprotein. This structure is called as a glycoprotein. Why it is called a glycoprotein? Glycoprotein means glyco means sugar, protein. When sugar and a protein combine, it forms glycoprotein. So this structure which is formed by covalent bond between sugar and a protein, it is called a glycoprotein. Similarly, just like a glycoprotein is formed, sometimes the head of the lipid, this is the head of the lipid, this is the tail. Sometimes the head of the lipid also combine with the sugar. These are the sugar I am showing. Sometimes the head of the lipid combine with sugar by covalent bond, and when lipid combines with sugar, it is called glycolipids. Glycolipid. So that's why two terms you have to remember: glycoprotein and a glycolipid. Protein plus sugar. So sugar plus protein. Sugar plus protein. When protein combines with sugar, glyco means sugar. Sugar combines with protein by covalent bond, glycoprotein. Similarly, when sugar combines with lipid, they combine by covalent bond, it forms glycolipid. So glycoprotein and glycolipid, these are the two chemicals which are only found on the outer surface of cell membrane. They are only found on the outer surface, not in the inner surface. They are only found in the outer surface of the cell membrane. Is it clear? They are not found in the inner surface. Okay. So this is all about chemical composition. So rest what we have discussed, 8%, 7 to 8% is filled by these carbohydrates molecules. And in addition to that carbohydrate, sometimes you find cholesterol, another chemical, cholesterol. So now I can show these are the oligosaccharides. These two are the oligosaccharide or sugar. I can write sugar, but the structure is not sugar. Complete structure is, this is glycolipid, this is glycoprotein. In addition to that, at regular interval, inside this phospholipid bilayer, you find cholesterol, which provides support. You find cholesterol also. Cholesterol is another type of fat or lipid. So cholesterol is also present inside the cell membrane. Cholesterol is a type of fat or lipid. It is also present in the cell membrane. So this is all about chemical composition of the cell membrane. But to explain how the cell membrane looks to a general people, this is the scientific explanation by Singer-Nicolson. When Singer and Nicolson given this model, Singer and Nicolson, two scientists, Singer and Nicolson given this model, many scientists and people unable to understand how this cell membrane is made of and what is the actual structure. So that's why, for clear understanding, Singer and Nicholson proposed a model, fluid mosaic model. Fluid. The model name is known as a fluid mosaic model. And this model is given just to compare with another structure present in our life. This is present in the cell membrane. But this type of structure, this cell membrane, is compared with a similar structure which we can easily compare. So, Singer Nicholson given a single sentence to explain this complete cell membrane. They told that the cell membrane is a fluid. Cell membrane is a fluid. Fluid means a liquid portion. And mosaic, inside that fluid is the mosaic means some patch are there. They given one sentence that cell membrane is nothing. It is a protein iceberg in a sea of lipids. According to Singer Nicholson, protein icebergs in a sea of lipids. This sentence is given by Singer Nicholson. Maximum time this sentence also comes as a question. According to Singer Nicholson, protein icebergs are present. Means protein are just like ice. Ice means a solid. Protein icebergs in a sea of lipids. Clear sentence. So sea of lipids. What they explained? They explained. Let it is a sea. This is a sea. Protein icebergs in a sea of lipids. I am explaining here. What is protein icebergs in a sea of lipids? Let this is a sea. Let sea is filled with what? Water. It is completely with water. Filled with water. Sea is filled with water. Okay. If in a water, ice piece will put ice piece or ice box like this. What will happen? The ice are solid. Yes or no? Ice is a solid, but water is liquid, in liquid state. When we put ice box in a water, the ice box can move inside the water. So that's why we can say protein ice box in water can move. Similarly, Singer Nicholson changed this into protein and lipids. They told the ice are nothing protein. Ice are protein and the water is made by lipids. So just like in sea water, ice can float. Ice piece can float in water. Now the protein can float in the lipids. So here water is replaced by lipids. You can see here, this is the lipid, black portion is lipid. So this portion is just like water. This portion is just like water. The black portion, this lipid portion is just like water, and these are just floating ice pieces, and they can float in both sides. Clear? Just for comparison, they told that the ice pieces are floating. It does not mean they will only float on the outer surface and the inner surface will remain free. Not like that. Both surfaces they can float, as well as they can bury inside. So that's why these are the icebergs, protein icebergs in a sea of lipid. Protein icebergs in a sea of lipid. According to Singer Nicholson, the protein are floating in the lipids, and these lipids which are present, this lipid represent a fluid part. As they are behaving as liquids, this represent a fluid part. So that's why Singer and Nicholson given this model into fluid mosaic model. Fluid mosaic. What is the meaning of fluid mosaic? The fluid part is created by lipids. The lipid is making the fluid part. This fluid part is made of lipids, and the mosaic means protein. Mosaic means a patch, and protein are present in patches. Protein are not continuous. You can see protein here, some gap, again protein here, gap, again protein here, like that. So protein are present in patches. So that's why these are the protein icebergs in a sea of lipid. In the sea of lipid, you can find protein icebergs. So this is called fluid mosaic model. But Seymour Nicholson told that we can compare this lipid with fluids, but this fluid is not complete water. This fluid, what we have written, fluid mosaic model, this fluid is not like complete water. It is a semi-fluid. Semi-fluid. Semi-fluid also called as a quasi-fluid. Quasi. Semi means quasi. So the fluid are quasi-fluid in nature. Quasi means just like a gel. Not complete fluid, not complete solid. So according to fluid mosaic model, the fluid, lipid part are making fluid, but these fluids are quasi-fluid or semi-fluid. We can compare with quasi-fluid or semi-fluid. One example I will give. Just you can compare with coconut water. During winter time, the coconut oil. The coconut oil, which is liquid at room temperature. During summer time, you find that oil is liquids. But if you compare with winter time the coconut oil gets settled and it form a semi-fluid like structure, gel-like structure. Not complete solid or not complete liquid, semi-fluid. So that is called semi-fluid. So here the phospholipid are semi-fluid in nature, and the proteins are present in mosaic in this phospholipid bilayer. And in addition to that, the cell membrane also contains 30 different enzymes. 30 enzymes. Just like inside our body, for digestion, different enzymes, digestive enzymes are there to break the food. Similarly, in the cell membrane, 30 different enzymes, 30 different enzymes are present in the cell membrane for different functions. We will discuss what are their functions, but you have to remember this detailed structure of plasma membrane according to fluid mosaic model. Okay, so another important part we have discussed in prokaryotic and eukaryotic. What is the difference between prokaryotic and eukaryotic? Many differences we have completed, but only one thing there is a similarity. If you compare a bacteria with human cell or bacteria with any eukaryotic cell, you find one similarity: the cell membrane. The cell membrane, there is no change. The cell membrane of prokaryotic is same as eukaryotic. Prokaryotic and eukaryotic cell membrane are similar in structure as well as function. So cell membrane never varies with respect to structure and function in both prokaryotic as well as eukaryotic cell. Okay, so this is all about cell membrane. Thank you. This much for today.